Our today's lecture is again on medical microbiology. Today, we'll see in detail Candida albicans, one pathogen which is included in your syllabus of MB 211. In this, our today's first part of Candida is based on its classification, morphological characteristics, cultural characteristics, its biochemical characteristics, as well as a pathogenicity. Again, we'll see certain predisposing factors, that is, conditions required for the diseases by the Candida. About this Candida, it is one yeast, that is unicellular fungi, and it is the most common cause of fungal infections worldwide. These are called as commensals also. Commensal, that is, they are normally present on our body, that is on skin, as well as they may present in the body, mainly in gut as well as in the genitourinary tract. Here, this organism, besides having characteristic of a commensals, may act as an opportunistic pathogen. Opportunistic pathogen, these are the microorganisms which are able to cause disease when conditions are appropriate for them. That is, whenever they get chance, they may cause a disease in human being. So this organism is commensal. At the same time, it is opportunistic pathogen. So here conditions appropriate for them means what? That is, when the mucosal barriers are disrupted because of several reasons, at that time this microorganism grow profusely in the body and cause disease. As well as, secondly, when immune status of that person lowers down, immune system if get compromised during that period also, they may invade and cause disease. These organisms are eukaryotes, unlike our initial organisms, pathogens included in your syllabus. They are prokaryotic in nature. E. coli, Salmonella, Staphylococci, these are all prokaryotes, while this pathogen is eukaryotic in nature. Now we'll see in detail classification of yeast. Classification of Candida. Now this is belonging to the kingdom fungi. Division Ascomycota, and class is Saccharomycetes. Their order is Saccharomycetes and family Saccharomycetaceae. And here the important genus belonging to this is Candida. Number of species are included, but here for us important one is albicans. So Candida albicans is main and important causative agent of several diseases of a human being caused by fungi. So among Candida species, as you know, this Candida albicans, which is a normal microbiota or a normal constituent of the human flora, may present on skin as a commensal. They are also present in gastrointestinal tract and genitourinary tract. They prefer that moist conditions present in the lining of this genitourinary tract. They are responsible for majority of candidal infections. So when they invade bloodstream, it is nothing but a systemic type of infection, and here to the disease name given candidemia. Here mia, that is presence of pathogen in the blood. And second possibility is they may invade tissues in the gastrointestinal tract, in the oral cavity, in the genitourinary tract. When they invade and when they grow, then it is called as a candidiasis. Another name given to the disease is moniliasis. So candidiasis, moniliasis, candidemia, these are variety of diseases caused by Candida albicans. Not necessarily every time Candida albicans is causative agent. Several other species they may involve in the infections. So which are they? Candida glabrata, Candida rugosa, Candida parapsilosis, and Candida tropicalis. With this, Candida dubliniensis and Candida auris recently identified species of a Candida, showing variety of infection in hospital environment. Mainly nosocomial infections, they occur because of this organism. Nosocomial infections are those infections which are acquired in the hospital environment from healthcare workers, hospital staff to the patient suffering from certain other diseases. So, very important role of this microorganism in the nosocomial infection, in the hospital bone infection. Recently, it is identified that up to 40% incidences of the infection by Candida, they occur in the hospital environment. So it is necessary to study this microorganism because of its different type of a nature, that is having commensal but at the same time opportunistic pathogen. About morphological characteristics of Candida. So as you know, these are unicellular fungi, that is yeast. You can see here, this is unicellular oval or a round structure of a Candida. Here, these cells are about 4 to 8 micron in size. That is quite larger if you compare it with the prokaryotes. They reproduce by budding. This is again a different type of a character. If you compare it with the bacteria, bacteria by binary fusion, they divide, while these are reproduced by budding. Here in this figure, you can see if this is the unicellular yeast cell, bud is formed in this way. Now you can see there is a continuous growth if appropriate conditions are present, and at the end, the number of a buds when formed, there is not detachment of this burst bud from the parent cell or from the first cell, because of which there is a formation of a such a filamentous like a structure. This is because of a incomplete separation of the yeast cells from each other, and this is called as a pseudohyphae. Pseudohyphae, pseudo means false, and hyphae, that is the filamentous elongated form of a structure. So here this is nothing but a dimorphic nature of the fungi. Dimorphic, di to morphic, morphology. Two types of a morphologies of organism observed, that is either they present in the yeast form or they may present in the pseudohyphal form. Sometimes true hyphae, because of a elongation of this microorganism, there is a formation of a hyphal form of a structures. And if you stain this microorganism using gram staining, they may appear gram positive. They are encapsulated, one thin slime may present, and these are diploid cells. Now, here they are in this way referred as a dimorphic fungus because of a two types of a morphology, yeast as well as a filamentous form. This one mainly observed on a primary media. Primary media, these are simple media. They are reduced nutritionally, and on that you can see budding of these elongated cells. And they are lined up in one line as they that particular bud is formed in one plane, and this mimic appearance of a hyphae, that is long filamentous elongated structure of a microorganism, microorganisms, and this is nothing but a pseudohyphae. And in some cases, you can see formation of a true hyphae also, but very rarely such a true hyphae observed. Basically, pseudohyphae is characteristic of a Candida albicans. Now, along the side of this pseudohyphae, false hyphae, there is a formation of a Blastoconadia. This one mainly observed around the septa. Septa, that is where they form particular bud. Only at that site there is a formation of a Blastoconadia. These Blastoconadia are nothing but a grape-like clusters. These are round, smaller grape-like clusters and present only at the separation point of two Candida albicans cells. Secondly, there is a formation of a refractile round structures at one end, at a terminal position of the pseudohyphae, and these are called as a chlamydospores. These chlamydospores are a thick-walled structures, and they are also observed when microorganisms grow in nutrient-rich media. So when you inoculate Candida albicans in corn meal agar, in corn milk broth with 5% of a milk, you can see there is a formation of a large number of a chlamydospores at one end of the cells. So here in this figure, you can see this one is a hyphae, pseudohyphae. Pseudohyphae, number of a candidal cells form elongated structures like this, and here where they form bud, that is now this one is the first cell, bud of which is this, bud of the second cell is this one, and here at the site where the bud is formed, where there is a septa, formation of a grape-like clusters. These are of a Blastoconadia. This is unique property of a Candida, that is formation of a Blastoconadia. At the same time, at terminal end at one end, mainly from the base part, when they present in cornmeal agar, there is a formation of such a thick walled structures. These are nothing but a chlamydospores. They are formed. So pseudohyphae, formation of a chlamydospores, and formation of a Blastoconadia. These are unique morphological characteristic of a Candida albicans. Some microorganisms like Candida tropicalis unable to form such a type of a structures, and so you can identify these microorganisms very well in the laboratory on the basis of such a type of a spores and a conidia formation. Secondly, about some important morphological characteristic of a Candida is that formation of a germ tube like this. So in this figure, you can see if this one is a unicellular oval structures of a Candida, if you inoculate them in appropriate media, that is cornmeal broth with 5% milk, there is a formation of a germ tube after 2 to 3 hours incubation at appropriate temperature, that is 37 to 40 to 45 degrees Celsius. So germ tube formation and the formation of a Blastoconadia, formation of a pseudohyphae and chlamydospores are used in the laboratory diagnosis of the candidal infections, that is to identify this microorganism in the laboratory. Now, here cultural characteristics of a Candida. So the most important media which is widely used for culturing this organism is Sabrod's dextrose agar. So this is Sabrod's dextrose agar, and here you can use any media having high percentage of a sugar for growth of a Candida. This one is used for primary isolation of organism from clinical samples. Means you can inoculate urine sample, blood sample, or whatever the scrapings from mouth where they grow profusely, and inoculate it on this sugar containing media, Sabrods dextrose agar, and see white colored colonies on this media. These are colonies having yeasty odor, and they are sometimes yellowish or a buff colored colonies on this media. These are quite large colonies. Nowadays, new culture media used by the researchers for cultivation of a candida, which observe mainly in hospital environment, that is use of a cornmeal agar. So here you can use in the liquid state, that is cornmeal broth with 5% of a milk, which is suitable for a formation of a chlamydospores. Then here after inoculation of it, inoculate it in that media, incubate it at 37 degrees Celsius in the incubator, and this is for up to 48 hours. And you can see massive growth of this organism in the form of a large white colored colonies. So here in this figure, you can see that buff white or yellowish colored colonies of a candida having a special appearance, that is the feet of that colonies extend out from the margins into the surrounding agar in this way. These are large colonies, opaque colonies, pasty, having yeasty odor, easily identifiable type of a colony characteristic of it. And after that, by using one method called as a wet mount method, one can identify this microorganism very well in the laboratory. So in the wet mount method, just prepare suspension of the colony and place it on clean grease-free glass slide. Put one cover slip on it, and unstained preparation you can easily observe under low power microscope. These are refractile circular oval cells, and if there is a formation of a pseudohyphae, you can easily identify that particular budding cells as well as a pseudohyphae under microscope. Next to which is the germ tube test. This one is again widely used in the identification of this microorganism. For this, requirement is of a human serum. So how to obtain human serum? So first collect a blood sample of the individual and not patient, because requirement is of a serum sample. Allow it to clot and the specimen, then place it in the test tube rack to speed up the clotting action. After clotting, then centrifuge it, and this is centrifugation at 3000 rpm for 10 minutes, and you'll get a clear solution, which is nothing but a serum. This serum now can be used for the germ tube test. In this, whatever colonies which appear on SDA, on Sabrods media, first pick up that colony and inoculate a looful of that particular colony into the test tube containing serum. So 0.5 ml of a human serum is sufficient. In that, inoculate your microorganism. Incubate it at 42 degrees Celsius for 2 to 3 hours, and then place one drop of mixture from this tube onto clean slide, and place one cover slip on it, and then see whether there is a germ tube formation or a not. This is called as a Brody's phenomenon, and one can identify this germ tube very easily within 2 to 3 hours. Now, after this, observe it under high power also for details of germ tube formation. Now, requirement is of a control is necessary. So, here control is identified or a known culture of a Candida albicans. This one available in laboratories. Number of that particular strain of Candida albicans is ATCC 10231, as well as second culture is required as a negative control is Candida tropicalis. As you know, Candida tropicalis unable to form germ tube. So ATCC 13803 is Candida tropicalis culture, and here use it as a negative control. Means simultaneously you have to inoculate two more tubes, one for positive control for Candida albicans and second serum containing tube for Candida tropicalis, so that you'll get correct result. That is how germ tube is formed in case of a Candida albicans, and how it is not formed in case of a Candida tropicalis, that is your negative control. So compare your test tube with Candida albicans and Candida tropicalis containing serum tube, and conclude or record your results. So this is part of a laboratory diagnosis of a disease caused by Candida albicans. Then for chlamydospores, as you know, requirement is of a cornmeal broth with 5% milk. Inoculate your organism in it, allow it to grow for 8 hours at 45 degrees Celsius, and here at one end of Pseudohyphae, you can see at terminal position round thick walled structures. These are nothing but a chlamydospores. So these are very important tests included not only in biochemical characteristics but also in the laboratory diagnosis of a diseases caused by Candida. Now, next part is of a studying pathogenicity of Candida albicans. So this organism have several virulence factors because of which it make them harmful for their host, and from that one and important factor is cell wall adhesins which are present which are produced by this microorganism. So adhesin proteins, they promote binding of organism to the host cells. As you know, this organism prefer mucous lining for their growth. So via hydrophobic interaction, with the help of these adhesins, they first tightly get adhere to that particular cell, and after that, because of such a type of adhesins formation, their attachment to the cells, it reduces the level of a yeast clearance from the body under normal immune regulation. Now, as you know, this one is a commensal organism. Normally, it is commensal, but it may be pathogenic under immunocompromised conditions in the individuals suffering from certain that type of a diseases like HIV, etc., HIV infection, that is AIDS, etc. Here, this Candida albicans penetrates host mucosal surfaces following morphogenesis. This one is morphogenesis. It changes its shape into invasive filament. Long filamentous structures are formed, and this pattern of formation of a filamentous pseudohyphae formation help the yeast to invade host tissues. Initially, only one single cell is present, and upon formation of a pseudohyphae, it will help this organism to penetrate into the body cells in the host tissues. At the same time, it produces various types of degradative enzymes in the body. These enzymes also help in the deep penetration of microorganism. So here, which types of enzymes produced by organism? Proteinases, aspartyl proteases, as well as phospholipids, which always help in the deeper penetrative type of entry of microorganism. And not only this, certain conditions of that person, body conditions, help in the spread of infection in the body. So to these factors, name given is predisposing factors or predisposing conditions. Means not necessarily every time upon entry of this organism, infection occur in the patient, in the person. Certain conditions which are already present in that particular body helps in the spread of disease in the person and convert it into the patient. So which are these conditions? First one will consider the category of a infants. Here bottle fed infants, they are prone to such a infections. Bottle fed, that is here for the feeding purpose when bottles are used because of a continuous irritation which occur in the mouth, a possibility of a infection by the Candida albicans, and there is a formation of one typical symptom called as a thrush in this bottle fed infants. This is oropharyngeal type of a infection and occur mainly in bottle fed infants. So feeding with bottle is nothing but a one predisposing factor. During pregnancy, chances of a infection of a urinary tract, they are possible. This is now ascending type of infection by the Candida. Then people who are overweight, it is also one type of a predisposing condition, as well as people with an underactive thyroid gland, hypothyroidism, one of factor, people with other inflammatory disorders, and most important one is prolonged use of antibiotics is important predisposing factor or a condition required for or appropriate for growth of a Candida albicans. See, in case of a genitourinary tract, the presence of a lactobacilli, which is beneficial microorganism, always help in maintaining pH of genitalia. Now, because of a prolonged antibiotic therapy, the level of or whatever the normal number of lactobacilli, it get reduced, because of which acidic products, they are also reduced and conditions are favorable for growth of a Candida. So vaginal infection, vaginosis, is mainly observed when level of a lactobacilli get reduced in the genitalia. Then uncontrolled diabetes. This is very important condition, that is diabetes mellitus. Here this organism prefer high level of a sugar for their growth, and as you know, during diabetes mellitus, level of sugar increases not only in the blood but also in the urine, and here this organism grow profusely in the presence of a high level of a sugar, and so urinary tract infections are very common in the patient suffering from diabetes. So it is one of important predisposing factor. Third one or next one is impaired immune system. Because of several reasons, immune system get disturbed. In that, role of a immunocompressed therapy is important. For certain autoimmune diseases, requirement of such a type of drugs, as well as during certain malignancies, that is cancers, requirement is of a immunosuppressed drugs, and during that condition body's immune power, immune systems get weakened, and now this organism grow very well in such a immunocompromised condition. Several diseases like AIDS, they also help this organism, that HIV, human immunodeficiency virus itself name indicate it attack our immune system, so conditions are now suitable for growth of Candida. Then irritation of a vagina. Irritation of a vagina, it may occur during other diseases also because of some other reasons, and again this organism grow in that condition. So people with weakened immune system, they prone to infection. People working in wet condition, this is again different type of a characteristic, as this organism prefer moist environment, that is having abundant moisture in their environment, they easily enter and cause infection. So this is all about predisposing factors. Now we can summarize this disease, that is Candida is nothing but one yeast unicellular fungi and it is most common cause of a fungal infection worldwide. Many species are commensals, that is, and as well as they are harmless, but some may act as a opportunistic pathogen. When there is some disturbance in the mucosal barriers, or if there is a problem to the immune system, that is, when immune system get compromised, they can invade and cause disease. They can grow very well in the laboratory on media containing high percentage of a sugar like Sabrod's dextrose agar, as well as on cornmeal media and on blood agar very well. Germ tube test is widely used for the identification of a Candida albicans in the laboratory. In the next lecture, we'll see detailed study of diseases caused by Candida albicans, as well as how to identify them in the laboratory, that is laboratory diagnosis, how to avoid the infections by Candida, that is prophylaxis, and treatment, as well as epidemiology, that is a distribution of the disease worldwide. So this is all about Candida albicans. Thank you. Thank you very much.